Recording. Okay. All right, now I got the recording going. Always make sure that I say I started the recording, please. All right, so we'll start going a little more in depth into the four supergroups, but it's primarily going to focus on what genus or species or general group falls under each supergroup. And I'll point out what I want you to focus on primarily. So for the excavates, we talked about it before. We talked about Giardia intestinalis, and it causes giardiasis. It includes protists with modified mitochondria, okay, which we hadn't gone over yet, as well as unique flagella. Now, as we said, some have a feeding groove or an excavate, but what I want you to focus on when we go into detail is that this supergroup excavates is divided up into three what they sometimes call subclades. Our diplomatids, our parabacillids, and our euglenozoles. So on this slide here, I want you to focus mainly on what we have here, which is what they have here also. Okay. Now going into a little bit of information on each of these subclades that make up the supergroups excavata. Okay, I talk about the first two together because the first two consist of organisms that have modified mitochondria that are either called mitosomes or hydrogenosomes. Okay, and these are our diplomatids and our parabacillids. Talked about Giardia intestinalis. It actually falls under this subclade, diplomatids, and it's said to have a modified mitochondria that's called a mitosome. Now, it generates energy like the mitochondria in our cells, except unlike the mitochondria in our cells, where it generates energy aerobically. These modified mitochondria in Giardia intestinalis and other diplomatids generate energy anaerobically. And this is comparable to our other subclade, parabacillids. Example of a parabacillid, Trichomonas Vaginalysis. This is a pathogen that causes an STD called trichomoniasis. But it also has a modified or what they call reduced mitochondria called a hydrogenosome. And it also generates energy like our mitochondria, but it's more closely related to the mitosome because it does it anaerobically. These are considered some of the earlier forms of mitochondria at one time that they used to refer to these groups of organisms that fell under diplomatids and parabacillids. They used to refer to them as amitochondrians, but now they actually refer to them as having modified. The third group, and these are just showing you some of the pics. The third group is the one you actually see hear about the most. These are your Euglenozoles. They're a diverse clade. They include predatory heterotrophs, well, heterotrophs are consumers, photosynthetic autotrophs, as well as parasites. Now, the main feature distinguishing them as a clade has to do with their flagella, which is said to have a spiral or crystalline rod that they don't know what the function of is it. Well, when I have students look at Euglenozoles, we focus on one of the two subgroups that falls under this subclade, Euglenids. But in actuality, this subclade is divided up into two smaller clades, kinetoplastids and euglenids. Now for kinetoplastids, what I mainly want you to know, okay, not the mitochondria part, but what I mainly want you to know is that they include free-living consumers of prokaryotes and freshwater marine and terrestrial ecosystems. And what else I want you to mainly know is what I gave y'all in lab. The most popular example of a kinetoplastid is Trypanosoma gambians, which causes African sleeping sickness, a neurological disease. That's the vector that transmits this is called the tsetse fly. And you can see here the Trypanosoma gambians is located. They show you a picture of somebody whose blood is infected. You can see the red blood cells, and you can see how these organisms are around them, and you can see the flagella on these organisms. Okay. But the ones that are most popular is the other sub or smaller subclade are euglenids. These consist of our Euglena, which you commonly see in teaching labs. But in actuality, what I want you to first know for euglenids is that they consist of autotrophs, heterotrophs, and mixotrophs. Again, Euglena is the most popular, but there's different species of Euglena, but this is the most common type that you see. That has chloroplast inside of it, has usually what appears to be a single flagellum and a red eye spot that helps it detect light. So like I said, there's a lot on the chart there when they show you how they divide up organisms into the four supergroups, or that polyphyletic tree, if you want to call it. But I just want you to focus on main key points. The second supergroup they call the Sarclade, which is made up of three clades. Our Staphylopods, our Alveolates, and our Riserians. Here they're showing you how they're divided up into their subclades, which is what we'll follow along with. So our Straminopoda includes important photoautotrophs as well as several clades of heterotrophs. Don't worry about this flagellum. I've been meaning to take it out. They say that's one of the trademarks, but we don't need that for us here. But next, what I want you to focus on for our Straminopods is that they're broken up into smaller groups. Our diatoms and our brown algae. Okay. Whenever you hear about plankton or phytoplankton, diatoms are one of two key sources that make up your plankton. The diatoms, though, they're considered unicellular algae, okay, with a unique two-part glass-like wall of hydrated silica. So when you look at diatoms, if you feel like you're looking at small pieces of glass, but these are actually living organisms. They just happen to have the same component that's used to make glass. They happen to have it in their cell wall, hydrated silica. The other group is our brown algae. Okay, they're the largest and most complex algae. Most of them are marine, and they're all multicellular. Okay, and they're one of three types of algae that are often referred to as seaweed. Now, the most popular form of brown algae, okay, are the giant kelp that you hear about off the coast of California. These are actually considered brown algae, so they're the most popular example that you see out of all of them. Is everybody else still can everybody else still hear me? Okay. Can somebody type on the chat board to check her connection? Would somebody type on the chat board to check her headset connection? Her mic, please. Thank you. Yeah, because we had that happen in another class, except this student kept typing in, they couldn't hear anything, and it ended up being that they didn't have their headset connected all the way in. So, oh, that reminds me, don't be surprised if we get cut off anyway. Okay. It is turning into one hell of a nightmarish adventure as we go through this semester. So right now what I'm finding out is Wednesdays are turning into horror shows for me. Something's always happening. If it's not with Blackboard, it's with Microsoft. So just to give you a heads up here. Okay. So the second clade second subclade for the Sarclade are our alveolates. This alveolate comes from alveoli. You ever heard of alveoli in reference to our lungs, our lungs that are tiny air sacs? Well, what they say is these organisms have membrane-bounded sacs. When you look just under their membrane, they have these tiny sacs. They don't know the purpose of these sacs, but this is where somebody came up with the alveolata for this clade. Now, what I want you to focus on is that they're broken up into three subclades or subgroups: dinoflagellates, apicomplexans, and ciliates. Dinoflagellates are the other group of organisms that are the major organisms that contribute to forming plankton and phytoplankton. Now, diatoms and dinoflagellates aren't the only ones, but they're two of the key components. Now, again, dinoflagellates, as you can see, it doesn't matter what clade or supergroup we're under, flagellata seem to be very common. But what I want you to focus on is they're the ones, okay, that are associated with causing this toxic red tides. The most popular dinoflagellate is Serratium, but it's not the only one. So this is a dinoflagellate also, and this is actually a pic of prepared specimens of dinoflagellates that my students would look at. Most of them have, if you're familiar with Star Trek and Klingon ships, that's how I describe it. Looks like a Klingon ship, excuse the nerdiness here. But most of them have this shape that we normally look at in a sample. And this is a subtype, a subspecies or a type of dinoflagellate called Serratium. But some of them are round in nature, so we have to point out to students that some of them are roundish, or have this little kind of a tail thing coming off of them. So I guess you could say there's diversity in their shapes, but the most popular one is Serratium. For apicomplexans, okay, these are parasites. Definitely want you to know there's parasites of animals. Some cause some serious human diseases, such as malaria. Okay, so Plasmodium. is a genus that's the best example of our apicomplexans. The most popular species of Plasmodium is falciparum that causes malaria. Okay, but there are actually up to 4 different species that fall under the genus Plasmodium that are associated with causing malaria in humans. And they are all spread by certain types of mosquitoes, and in humans the parasite can be found mainly inside of the red blood cells. And unlike Trypanosoma gambians, okay, and the tsetse fly being the vector, here it's a mosquito that's the vector. And unlike Trypanosoma gambians, where yes, you end up with the organism when you get infected in your blood, the Trypanosoma gambians doesn't get into the red blood cells. For Plasmodiums, all 4 species, and in this case falciparum, it actually gets into the red blood cells, okay, and uses the red blood cell to help it continue its life cycle. Okay, now the last group are our ciliates. Okay, they are a large, varied group of protists and are named for the use of their cilia to move and feed. The 2 most popular types, and we often use these in teaching labs, are Paramecium and Stentor. Now, if you had 1406, you definitely got to see Paramecium if you were in class in the spring before we went on break. Sorry, everybody, I was trying to write something down here. What I keep forgetting to move to my other PowerPoints is this is a more exact pic of Paramecium. It doesn't look like this under the microscope. It looks more like this or this under the microscope, but actually it's a hairy little guy. It's covered with cilia. Okay, it has structures such as an oral groove. It has vacuoles such as contractile water vacuoles here. Whatchamacallit, but it's covered in cilia. And this is the guy that moves really fast. It's a lot larger than Euglena, but it's definitely very popular cilia. I'm sorry to go back a little bit here. Normally you can see the outer edges of the cilia. If you adjust the lighting and the iris diaphragm in the scope, you can see the cilia. And I don't have Stentor. I thought I had a picture of Stentor. Stentor, the best way to describe Stentor, okay, is I call it the kind of the elephant of the protist world that we look at. It can round up or it can stretch out like one of those bugle horns. When it rounds up, it looks like it's got a single eyeball with this little grin on its on the side, like if it's grinning at you. And that area, that grin area, is roughly in the mouth area, and you can see the cilia along that area. And so it also uses its cilia to help it move around, in addition to the fact it can actually change its overall body shape. I forgot to put one in. Okay. The last subclade of this supergroup, the Sarclade, are the Riserians. Okay. The Riserians include amoebas. If you recall, I said for amoebas, what we look at in the teaching lab is often an amoeba called under the genus Chaos, and it falls under the supergroup Uniconta. But there are other amoebas that fall under the Sarclade, specifically the subclade Riserians, or this right here. So if you look here, though, first of all, Riserians, what I want you to know, in addition to this amoebas part, is that they're broken up into 3 groups. Okay, they consist of our radiolarians, our foraminiferans, and our cercozoans. The radiolarians, I do want you to notice they have pseudopodia to engulf microorganisms through phagocytosis. Okay, but those pseudopodia radiate from a central body. This is an example right here. Okay, how it can radiate out. If you've ever seen the amoeba that they often show in the teaching lab, and that like the one I show in the teaching lab in 06, it doesn't look anything like this one here. Now for our foraminiferans, they often go by forams. Okay, and they get their name from their porous, generally multi-chambered shells. They call these porous, multi-chambered cells tests. Okay, now they will extend their pseudopodia out through these pores. But when I would show students prepared slides, okay, this is what they would see. So on the prepared slides, you really don't get, an exact idea of what you're looking at. It feels like you're just looking at pieces of glass as you're looking at it. The last group are our cercozoans. These include most amoeboid and flagellated protists with thread-like pseudopodia. They live in marine, freshwater, and soil ecosystems. Now for these, the cercozoans, I'm still trying to confirm, okay, if you're familiar with the brain-eating amoeba, Infowlary, still trying to get down, find out where exactly it's being reclassified under. It's right now under Kingdom Protist and the phylum Percolozoa. Don't quote me on that, but I think it's called Percolozoa. But it's being reclassified, and right now, big question mark. This is that amoeba that causes the brain-eating amoeba that's associated with still water in lakes that people get infected and it burrows through your nasal passages. I don't know if it's actually one of the amoebas that falls under Sarclade, but I put the picture there because so far that's what I can find. Okay, good. All right, good, Chanel. I'm sorry, I had this happen to another student a couple of weeks ago. In their case, they're using a headset that they got to connect to their computer, and they didn't push it in all the way. So, but the problem with that is they kept assuming that it was on my end, and every other student kept saying they could hear. But if it's not one thing or another. Welcome to online. Good to have you though. Okay, we're going to move into the next supergroup. Okay, so make sure you listen to the recording and listen to what I'm pointing out for you to focus on the test, because there are little tidbits here and there I'm not going to focus on. So I'm trying to point out what exactly here to narrow it down. Now, Archaeplastida is our third supergroup. Archaeplastida is broken up into three subgroups. Okay, our red algae, our green algae, and then our land plants. Now, we're not going to talk about land plants here because there's 2 whole chapters devoted to land plants, 29 and 30, so we'll visit that later. We'll focus on our red algae and our green algae, okay? The red algae, well, they're reddish in color due to an accessory pigment called phycoerythrin, yes, I want you to know that, which masks the green color of chlorophyll. Some of these pigments, it's kind of like with the halobacterium that fall under the domain Archaea. You know, they give the Great Lakes a little pink tint, but it's because they have a red light-absorbing pigment in their membrane. But that red pigment, light-absorbing pigment, doesn't function the same way as chlorophyll. It actually protects those archaeans from UV lights from the sun. So these pigments can have different functions. Chlorophyll obviously involved in photosynthesis. And red algae are multicellular, and they are the largest. I'm sorry, they are not the largest. That's the brown kelp, the brown algae. But the largest are considered seaweeds. And if you recall, these red algae were the ones that were considered the first, or it was a species of red algae that was considered the first multi-eukaryotic organism to evolve based on fossil data. Now, green algae, they are named for their grass green chloroplasts. They can be unicellular or they can be multicellular. Keep that in mind. Plants are considered to be descended from green algae. Okay, so we'll talk about this more when we get into chapter 29, and we'll mention this group here, charophytes, and we'll talk about some homologous traits between these two that tie them into being it's from green algae. We already talked about before for the red and green algae. It's tied into what eventually we call secondary endosymbiosis, about how over 1 billion years ago a heterotrophic protist acquired a cyanobacterial endosymbiote, which ends up being our chloroplast. This photosynthetic eukaryotic descendant of this ancient protist is believed to have evolved into red and our green algae because something happened to that symbiote, that cyanobacterium, that led to this diversification that occurs. And then down the line, they're still trying to fill in the gaps to provide further evidence that land plants are descendants of green algae. And this is just showing you some pictures of these guys here. But for the green algae here, in our teaching lab, especially for 1406, students will often look at green algae, Clostridium, which is a unicellular green algae, as well as Volvox, which is a colonial green algae. So for red algae, we describe them as being multicellular. But for green algae, what I want you to know is there's more diversity. They're unicellular, colonial, and multicellular. Okay, so they're very diverse. The last group is our Unicops. And I'm sorry if I'm talking so fast. I just hoping I can get through this before anything else disastrous happens, okay? Forgive me, all right. But the Unicops, okay, they include protists that are closely related to fungi and animals. And many of them fall under amoeba or Amoeba Zones. So when we take this supergroup, the Unicops, we first divide it into 2 clades. Our Amoeba Zones, okay, which is what they show you here, and our opisthecons, which is what they show you here. So on these slides, if you look at that initial slide where I show you all 4 supergroups, Then what I show you is as we go into detail with each supergroup, I'm only showing you part of that chart. Okay. Now the way it works for the amoeba zones, as they show you here, they're divided up into the slime molds, tubulinids, and into amoebas. Okay, so basically amoebas, thus the amoeba zones. Notice the opisthecons though. They're divided up into our animals, our fungi, and then some protists known as choanoflagellates. Okay. Animals we'll visit later. We have 3 whole chapters, actually 4 technically if I get into the last one. Fungi is 1 chapter. That'll be the first chapter we'll actually go into, which is actually chapter 31. We'll jump over plants initially and go into fungi and then come back. This is what your lab is focusing on today. But going into amoeba zones. Amoeba zones, well, they're amoeba that have lobe and tube shaped rather than thread-like pseudopodia. So as I said, normally we look at chaos. That's the genus of amoeba that we look at, and it falls under these amoeba zones. Now they include the slime molds or tubulinids and entamoebas. Now for slime molds, they were once thought to be fungi, but molecular systematics has now placed slime molds in the clade amoeba zones. They have diverged into 2 types. Now, I forgot to put the link on here, but did anybody happen to see the PBS documentary on plasmodial slime molds, or none of y'all have reached that point to where you really care to see those? Okay. I just thought I'd ask. Okay. That was a definite no. All right. Yeah. No, I haven't put it, Catherine. I'm going to try to find it. I forgot to see if it's available from PBS so I can add the link, okay? I'll find it. So basically, for the plasmodial slime molds, they're described as multinucleated, but they're unicellular organisms. And I'll try to find the video link. PBS is getting a little strict on some things. Oh, yeah. I try to get around it. There was another video link I gave you, Life's Rocky Start. I had to get around it because the original link, it cut me off, and it started telling me I had to log in and everything. Yeah, and I'll try to get it in there if I can, Catherine and Elizabeth, okay? If I can get around it, if I can. But yeah, I'm used to them making it easy to get the videos. It was just an issue of how long they would be available, not an issue of, I've got to jump through hoops to get there. But it was on plasmodial slime molds, and this is just a little side note. It was interesting. They were doing research, and the research they conducted basically demonstrated that. Let me show you the organism. The organism looks like this. Okay, doesn't look like one organism. First of all, second of all, it doesn't look like an organism to most. But basically, what they said is, don't be deceived by its looks. So far, this organism is demonstrating it knows what it's doing. Okay. Basically, what happens is it grows in the direction of food source. Okay, so this decaying tree here, it's growing over it, and what it's doing is breaking it down. It's a decomposer, absorbing the nutrients, and thus it'll grow larger and larger. But it looks like almost like a net, a colorful net, but it's one organism. It has one continuous plasma membrane, but it's not a perfectly round plasma membrane, but it has many nuclei inside of it. And they did some studies such as altering the surface that it would substrate or surface that it would have to grow across, and they found it would actually adjust, okay, how it would approach that surface. It would either slowly grow over it or go around it. Now, what was making it grow or go around it was a food source that was placed, say, 12 inches away from it. It knew to grow towards the food source. Well, the way it works in lab is Joel, or my lab tech, will give a petri dish, and it'll have these little oats from oatmeal. They'll be spread out roughly about a ½ inch to an inch apart. And the plasmodium, basically, it looks like it starts off in the middle of the petri dish, and it grows out towards those flakes. It's able to detect them. So in the video, what they ended up doing was comparing this. Have you all ever heard of the movie The Blob? Probably going to get nothing again, but have you all ever heard of a movie called The Blob? Okay, if you haven't, look it up. Okay, thank you, Joey. One of y'all have. Look it up. They were basically comparing it to The Blob. Thank you, Zoma. At least somewhere in here, y'all understand what I'm talking about. Now, there's another type of slime mold called cellular slime molds, which are multicellular. But going into these, if I can find the link, they're going to give you way more, and it may not agree 100% with what I have here, but this is general information. Now, there are said to be many species of slime molds. Now, in the video, they just talk about them as if there is only one. They're usually yellow or orange. At one point in the life cycle, the plasmodial slime mold forms a mass called a plasmodium. Okay, not to be confused with the genus Plasmodium that causes malaria. What they say, Plasmodium is not multicellular, even though it looks like, if it's like this, how could it be one single cell? But it is. And it is undivided by the plasma membrane, but it contains many diploid nuclei. As I said before, it'll extend its pseudopodia, so these are considered pseudopodia, through decomposing material engulfing food through phagocytosis. And as I said, it has a way to pick up on food source. Okay, it doesn't have to be, how do you say, placed on it. It can actually go over a substrate that's not considered a food source to reach its food source. But when it goes over a substrate or a surface, it actually can pick up on what's on that surface and might cause it to adjust how it moves towards a food source. For the cellular slime mold, I just have something very quick. It's multicellular and it goes through 3 life cycle stages, actually: individual, colony, and spore. They didn't talk about this one on the video, so I don't know if the information is changing or not. I'm basically going by the text, but this is the best pick I could find for the cellular one, more clumped up. The more popular one, again, is this one, the plasmodial slime mold. Okay, the remaining 2 groups under the Amoeba Zones are the tubulinids and the entamoebas. Now, the tubulinids are the common unicellular Amoeba Zones in soil, as well as freshwater and marine environments. Most gymnomoebas are heterotrophic and actively seek and consume bacteria and other protists. Now, like I said, in the teaching lab, we usually look at an amoeba that falls into the genus Chaos, and Chaos is said to fall under tubulinids. So that's your classical amoeba that you see in the lab. But there's entamoebas, and these are parasites of vertebrates and some invertebrates. The most popular one, for not a good reason, is the entamoeba histolytica, because it causes amoebic dysentery, the 3rd leading cause of human death due to eukaryotic parasites. Okay, and basically it's associated, again, like with Giardia intestinalis, it's mainly associated with 3rd world nations. It targets the intestines, and causing diarrhea, but also may contain blood and mucus, so it can get pretty serious here. And you can see many prokaryotes and eukaryotes, that's one of the symptoms that's going to end up being. So here it is here. This would be an example of chaos, what we look at in the lab mostly. But this is the entamoeba. You can see all amoebas don't look this way. They have various shapes. They're very diverse in their shapes, as well as they fall under 2 separate supergroups. Now I'm going to transition into the last part, okay? Like we did with prokaryotes, we'll talk about the roles protists play, the ecological roles. So it says protists play key roles in the ecological communities. You can change that to the biosphere like we have for prokaryotes. And I'm going to give you 4 key roles, okay? First, they're found in diverse aquatic environments. And what I left off here is those such as dinoflagellates and diatoms. They're 2 main sources that make up plankton that many organisms, both fish and mammals, feed on. Protists often play a role of symbiote or producer. Some symbiotic protists benefit their host. Photosynthetic dinoflagellates, again, are food-producing symbiotic partners of coral polyps that produce coral. So here, in this case, they're not even acting as plankton. Okay, they're performing a symbiotic mutual relationship in this case. And then some protists are wood-dieting protists. They're capable of breaking down cellulose in the guts of termites, along with your methanogens. Okay, so they make up part of your microbial flora. It's not all prokaryotes, or it's not all bacteria, in other words, your microbial flora, even though they get the top billing. And then some are parasitic. Okay, yeah, that's a role. That's a role in the ecological community. It's not all positive. We already talked about Plasmodium falciparum being one of 4 species that contributes to malaria, this being the most common one. But there's also Phytophthora. Okay, I'm not even going to try anymore. Just do the genus that causes sudden oak death also. That's actually caused by a protist. Okay, not a fungi or a prokaryote. And this is just showing you here a symbiotic protist. This organism is a hypermastogote, a member of the group of parabacillids that lives in the guts of termites. So they've actually isolated one and gotten a pretty good picture from your textbook, or from one of the textbooks. I don't remember if it was the 11th or one of the older editions. So it looks like Cousin It from the Addams Family. I don't know if y'all heard that one before, but that's what it makes me think of. Hairy little guy. The fourth role is some protists are photosynthetic protists. Okay, many protists are important producers that obtain energy from the sun and form the base of the ecological food webs. In aquatic environments, photosynthetic protists and prokaryotes are the main producers that all organisms in the community depend on. So again, when you hear talk about plankton or phytoplankton, yeah, you can have your prokaryotes contributing to it, but you have your dinoflagellates, you have your diatoms, you have green algae also contributing to it. All of these contribute to the plankton. That's a key source for marine environments to survive on. So in aquatic environments, photosynthetic protists, okay, are limited by their low levels of nitrogen, phosphorus, and iron. These populations can explode when there's an increase in these elements. And this happens through human actions, such as using certain insecticides or pesticides, or just fertilizer for growth sometimes. That's a key one, actually. This can alter the populations of other species in the community. But here it's just showing you that interconnection, okay, between marine life, okay, and here you have our protists that fall under these groups, and it's just showing you the relationship they have. Now, with global warming, okay, how will global warming affect photosynthetic protists? Well, the biomass of photosynthetic protists has declined as the sea surface temperatures have increased. So these protists that contribute to what I refer to as our plankton in general, they're found more towards the surface, and as the sea surface, or the surface temp of water increases, okay, this interferes with their growth. Well, if you look here, how does the rising sea surface temperature reduce the growth? It has to do with the interference of nutrient recycling. Okay. The belief is that warm surface temp prevent cold nutrient-rich waters from below from rising to the top. So it's interfering with the cycle of water, cold water, rising to the top. And when it rises to the top in our oceans, it draws up a lot of the nutrients to support growth of our protists. With the rising temperatures affecting the surface temp of water, that's interfering with that cycle, so there's less nutrients towards the surface. If the ocean surface temperatures continue to warm due to global warming, this could have a large effect on marine ecosystems, fishery yields, and the global carbon cycle. This is what this is supposed to show you, okay, how various living organisms are interconnected with each other, okay? So what we don't see with the naked eye, okay, is extremely important to what we do see with the naked eye. So we realize it now or not, we will realize it eventually. The effects on this. I think have most of y'all heard of what they used to call NOAA, but now weathermen and weatherwomen like to say NOAA. Have y'all heard of this? What it's called NOAA, National Oceanic Atmospheric Administration. Yeah. So what they've been doing is they've been monitoring global temps, okay? And when they monitor global temps, they also monitor sea surface temps, such as SST. And they just put a little chart together yearly. And I forgot to check to see to make sure this video is working, but check this link for this. And they just monitor this as well as overall global temps, such as 2016. We keep hearing this is the hottest year, this is the hottest year, and it seems like, okay, well, each year is the hottest year. Here's a link if you're interested in. It should still work and take you to NOAA. I'm not going to test you on any of this information that has to do with NOAA, but I put another one up from 2019, and it just shows you the changes in temps. But for these graphs, it's showing you the changes in temps, not just sea surface, but also land temps. It's just showing you these constant adjustments. And then these are some information that they had for 2016 as far as selected significant climate anomalies and events in 2016 that they recorded, and they want to know, is it an anomaly? Hey, okay, it won't happen again, or is this a sign of things to come? And then I put one up for 2019 if you're interested. And then this was just some information off of 2016 because you heard how we pulled out of the what was the name of the pact we had to try to regulate global warming by 1 to 2 degrees, which was not working. We pulled out of it. Yes, the Paris Accord. Thank you. God, that's bad. I'm forgetting everything. Thank you, Chanel. I'm glad you could hear me now. But yeah, so this is just some information. Now, obviously, we're in 2020, not too far off from 2021, but just take a look at it. If not, at the very least, go to this site. Again, they're referring to it as NOAA, but it's NOAA, and hopefully I said it correctly. National Oceanic Atmospheric Atmospheric Administration. There you go. Double check myself. It's a good site to check into if you want to know if you're being fed a line of bull or if there's actually some evidence to support it. Now, I thought I put it on here and I didn't. Maybe I put it on earlier. I think maybe I might have put it on chapter 25. I thought I put a link to an article about the volcanic activity in Hawaii as the lava was moving across the surface, the land, and into the ocean. It was grinding up and churning up the soil, okay, which meant that it was pulling up on nutrients in the soil and dragging it into the ocean. There was an article that talked about how there was what they referred to as a phytoplankton explosion. In other words, in general, plankton itself, or the organisms that make up plankton, such as these, such as these, such as these, and such as these, there was suddenly a population explosion, a growth. Okay, so even though this the surface temp is increasing, affecting the cycling of water from lower depths, which are colder water of colder temps, and thus interfering with the pulling up of nutrients to the surface to support plankton growth. Okay, there are still other factors that, at least in regional areas, might be able to help preserve and keep numbers up for these smaller organisms that make up our plankton, which are both of prokaryotic and protist nature. Okay, such as lava flows that go into the ocean, dragging in nutrients. Okay, everybody, I did not, honest to God, I did not think we were going to make it through this. I'm having trouble with my own computer. Let me stop the recording first. Or actually, before I stop the recording, let me mention the test. The test is going to be the same format, 50 multiple choice questions. On Tuesday, make sure you log in a couple of minutes before 12:30. At about 12:30, under the folder exam, you'll have exam 2 appear. It's the same procedure as last time, okay? Just make sure that you understand, just like before, you're responsible for knowing what you need to have a good connection to get in there and take the test. It'll take one heck of a miracle to grant a makeup. I mean that seriously. I know that there are problems that are occurring, but we've been in this long enough. We're going to be in week 8 when you take the second exam, so it's a 16-week semester. So just make sure that if you happen to be having any internet connection problems, make sure you try to go to one of the ACC campuses. Look at the information I gave in the PowerPoint and syllabus, but don't take any chances, please. But it's the same format, 50 multiple choice questions. You have an hour and 20 minutes to do it. The exam will appear at 12:30. Just complete the answer, submit it. If you're not through in an hour and 20 minutes, it'll auto-submit automatically for you, and that'll be the end of the test, and it should show you grade pretty immediately. And then also, again, I don't talk much about the answers, if at all, because we are still staying online, everybody. We are staying online through the spring. That's already been confirmed. We're already assigning classes. So if you haven't heard that and you're going to stay at ACC or think you might take classes, be aware of that also. Okay? Let me stop the recording.